Okay. So let's start with what sequences to use. Well, first of all, how does this process work in general? What you're doing is you're taking the similarity between sets of sequences and looking to see how similar they are to each other and making the assumption that the more similar they are, the more recent their last common ancestor. So you're looking for a molecular clock. And so here's the notion here, is if you have two sequences, two pairs of sequences, and one pair are a lot more similar to each other than the other, the assumption is that they diverge from each other at different times, and that's reflected. The notion is that when this last common ancestor was, where one organism became two, and they each went their evolutionary paths, that the sequences continue to drift at random, and so the more drift there was, the less similar they are, and that represents a deeper ancestor. This is true more for some molecules than others, and the identification of a good molecular clock is about making sure your molecule follows this rule. And there are many, many cases where it does not, or where you can't see it. So what are the properties of a molecule that you're looking for? They have to have clock-like behavior. What that means is that the sequence has to be long enough to contain data. You can't look at, so for example, transfer RNAs are terrible molecular clocks because they're only about 80 nucleotides long, and half the nucleotides are conserved. You really only have a handful of nucleotides you're looking at. And so the statistical stochastic variation in the sequences drowns out any real phylogenetic relationship. It has to be long enough to be statistically meaningful. The bits have to evolve independently. And so this is one of the shortfalls of protein sequences for phylogenetic analysis, in that a change in an amino acid in the hydrophobic core of a protein has an impact on all the amino acids around it, right? Because it has to be space filling. They have to pack together in that hydrophobic core. And what that means is that the residues can't really evolve independently. When one changes, that puts pressure on all the others. Now that's also not true of RNAs, right? Because most RNAs are made up of base pairs, and when one nucleotide changes, the other does too, or it puts pressure on it. But every base pair is an independent unit. And so RNAs are made up of lots of little bits of information that can change independently. You have to have an appropriate amount of sequence variation. Going back to the molecular clock notion, you wouldn't try to measure your lifespan with a stopwatch. Right? That doesn't make sense. If you have a sequence that varies really, really fastly, that evolves very quickly, that's a terrible molecule to study deep evolutionary relationships. And likewise, if you've got a very, very highly conserved molecule, you can't use it to study close relatives because the sequences are going to be the same, or so small a number of differences that there's no statistical validity to try to measure that. They have to have an appropriate phylogenetic range. This is a very fancy name for something that's very trivial. Let me give you an example. Cytochrome oxidase is a terrible molecular clock to study bacteria in general, because about half of bacteria don't have cytochrome oxidase. And you don't learn anything about Clostridium looking at cytochrome oxidase, right? You can't include it in the tree. But it's more than that. A big part of this is gene families. And so not only does the same gene have to be in all the organisms, but the same kind, it has to have the same function, has to have the same role, and in the case of multi-gene families, it has to be the same homologue. Gene families are a nightmare for this kind of thing. And you can imagine, I'm sure, creating a phylogenetic tree of mammals using globin genes. Mammals have tons of globin genes. They've got several versions of myoglobin. They've got a hemoglobin A and B, alpha beta. I mean, they've got fetal alpha globin A, adult alpha globin A. They've got globin Bs. I mean, there's just a giant gene family. And if you want to create a tree based on these, first of all, you've got to pick one version, right? Alpha hemoglobin A. alpha hemoglobin, fetal alpha hemoglobin. And you've got to use that fetal alpha globin in all cases so that you're not creating a tree of the globin genes, you're creating a tree of the organisms. But all the organisms have to have that same kind of gene, that alpha fetal hemoglobin. And not all mammals do. You don't want horizontal transfer. The classic example of this is antibiotic resistance genes, right, which move around in bacteria all the time. You can create a perfectly reasonable tree, for example, of the neomycin resistance gene. And that tree will be valid. It'll be a perfectly useful gene to study the evolutionary history of neomycin resistance, but it will tell you nothing about the organisms you happen to have fished the genes out of. Finding genes in which horizontal transfer is unlikely is not as easy as you might think. You have to have sequence information. If you're going to use some obscure gene, you may have to do a bunch of sequencing yourself, right? Instead of being able to go and fish your sequences out of GenBank. It's a lot more convenient if you've got all the sequences you need at hand already. Particularly if they're big, long genes, or if you're going to be doing different kinds of genes and this sort of thing. You also would like some other resources. There's some really interesting resources online for some genes and not others. And so when you put all these things together, what you find is there is a clear standout for the best gene for this kind of analysis, if you're going to use a single gene. And that's the RNA of the small subunit of the ribosome. I call it the SSUR RNA. I may slip sometimes and call it the 16S RNA because it's in bacteria. But I'll try not to. So why is a small subunit of the ribosomal RNA the best? Number one, it's present in all living cells. That's a big one, right? It has the same function in all cells. And as nearly as we can tell, it really is exactly the same function. It's quite common even for these RNAs to be interchangeable. You can take the small subunit RNA from one organism, put it in another, and it will assemble functional ribosomes. Now, this actually is the weakest of these because there are functions of the RNA that are a little different in different organisms. And this is inevitable. The classic example of this is the temperature that it's designed to function in. Thermophilic RNAs are different than mesophilic RNAs, and that shows up as an artifact in trees if you're not careful. So the functions aren't identical in any gene, but in terms of the best, the ribosomal RNAs are that. It's big, it contains 1,500 to 2,000 residues, most of which evolve as pairs, base pairs. Big enough to be statistically useful, but not so big that it's a hassle to sequence if you have to do it yourself. This is why this large subunit of the ribosome hasn't been used as much, because 3,000 nucleotides, it's a lot of work to get the sequence all the way through. There are some other reasons that have to do with clock-like behavior and so forth. It's made up, this is what it looks like, it's made up of about 50 independently evolving helices and about 500 independently evolving base pairs. So essentially there's 500 data points in this RNA that are useful for phylogenetic analysis. It's conserved in sequence and structure, and that means it's easily aligned. I don't know if I've got, this happens to be E. coli, but I showed you last time with human and E. coli next to each other, and they have the same secondary structure. And the structures of RNAs make them really useful for alignment. And I'll show you that in a little bit. Yeah?
Let's see this.
Circa, approximately. The really interesting aspect of this RNA is that it contains some bits that evolve very, very quickly. These are the second hand of the molecular clock. And there are other bits that are very, very highly conserved. These are the hour hands. And so, for example, this piece of the E. coli RNA is actually on the surface, and it doesn't interact with any proteins, and it doesn't have any direct function in translation. And so this can evolve very quickly. As long as it stays out of the way, it can do almost anything at once. And so this is a second hand. This changes quickly. This stuff in here, for example, is right at the catalytic center of the RNA. Actually, since this is a small subunit, it's where the messenger RNA and the codon-anticodon interaction is measured. And these things are very, very highly conserved. And so these are the hour hands or even the clock under water. And everything in between. And so one molecule can be used to study relationships that are similar between related organisms or very distantly related organisms. The range of this variation is more than is the case for the large subunit RNA, which is one of the other reasons why this has been used. Do you have a question?
How do you separate convergent evolution from divergent evolution?
So that's an interesting question. And convergent versus divergent evolution. The reality is that in a sequence this large, 1,500 nucleotides, convergent evolution is astonishingly unlikely. The number of possible sequences, the number of possible functional sequences is quite literally astronomical. 10 to the 69 or something like that. There are more possible sequences of ribosomal RNA than there are atoms in the universe. And the result is convergence of those sequences is really unlikely. Except, I talked about high temperature RNAs. There is a kind of convergence that happens there where molecules can converge on GC richness, for example. And that's actually more complicated than I suggest here. And so that's a kind of convergence that can show up as an artifact of the structure. But that's true for proteins as well and for other RNAs as well. Horizontal transfer happens for any gene, but seems to be a lot less common for genes that are involved in the central information processing of an organism. Think of the central dogma. As opposed to metabolic functions, which are transferred readily. And the ribosomal RNAs seem to be more resistant than anything else. And this makes sense when you think about it, because this RNA has to interact with lots and lots of stuff. And the probability that an RNA could move from one organism to another and then appropriately interact with those things in such a way that the organism remains competitive and can survive in the long run is pretty unlikely. That being said, there are some cases known for horizontal transfer even of ribosomal RNAs. In some cases you can see that directly, in some cases you have to refer. Lastly, there are huge databases of sequences and sequence tools that are really easy to use, and those of you in the lab will get to use that as the current project. Here's a secondary structure. Another aspect of deciding what sequences to use is deciding what organisms to include, right? Okay, you've decided on the ribosomal RNA, but what organisms do you include in the tree? Generally speaking, this is something you don't decide in advance. You've got probably an organism of interest that you want to know about. That obviously is in there. And then typically what you'll do is you'll iteratively build trees, have a look at it, think about what it means, pull sequences out that don't add information, add new ones in, and repeat the process over and over again until you're happy with the tree. You have to have a good outgroup, and sometimes that changes over time. We'll talk about all these things as it goes along. So I don't want to dwell on that.